Okay, let's get started. Somebody asked, is it okay to have just everything for your unknown in a PowerPoint? And the answer is absolutely. In fact, that would be my preference instead of having multiple documents. Is that everything is kind of compact enough that you can list discrepancies on a PowerPoint slide. You can list, I know some of you have taken pictures of some of the keys you use and things like that. So everything in a PowerPoint is fine, in fact, preferable. Any questions?
Do you want two separate PowerPoints, one of the unknown and one of the known?
Yes, I would like two PowerPoints. I would like one of your Nolan pictures for 14, 15, 16, and then a separate document for your unknown project. Okay. I was talking to Dr. Morton McCombs on the way in this morning, and I said I felt a little weird. This is my last fall semester lecture. Dr. Rhodes and I will be splitting a job starting the next 2 years. So he'll teach in this spring or fall, and I'll teach in the spring. So kind of strange. Okay. What we need to wrap up today is to talk about a couple of more serological tests. And then whatever time we have remaining, we will talk a little bit about monoclonal antibodies, which are becoming the basis of a lot of the serological tests that we're using. So last time, I said this was a great point that we could talk about the ELISA test. ELISA is an acronym. What does the E stand for? Enzyme, yes, we have worked with these ELISAs before. ELISA stands for enzyme-linked immunosorbent assay. So the key elements there is that there's going to be an enzyme involved in this particular reaction. And there are going to be antibodies involved in this particular reaction. Immunosorbent, immunology, antibodies. The power of ELISA is that it is a highly sensitive testing method. So much so that theoretically you could detect a single antibody molecule if you're screening for antibody, or a single antigen molecule if you are screening for antigen. There are two categories of ELISA assays. There is direct and there is indirect. And they're screening for different things. Direct ELISA methods are looking for the presence of a particular antibody. Indirect are looking for the presence of a particular antigen. Indirect is also sometimes called capture or sandwich method. And when we look at those two procedures in just a second, you'll see why that's the case. So, what's the enzyme that's involved in this process? It's a phosphatase enzyme. You know what a phosphatase enzyme does? Must have something to do with phosphate groups, right? A phosphatase group is an enzyme that lops off a phosphate from a molecule. And typically, the substrate that's used in ELISA reactions is a compound called PNPP. I don't really care that you know the name of the acronym, but this stands for para-nitrophenol phosphate. PNP stands for para-nitrophenol. Okay? So the phosphatase enzyme goes, boop, knocks off that phosphate. PNPP in solution is colorless. PNP, paranitrophenol, is yellow. So we can detect the presence of a phosphatase enzyme by incubating it with substrate and see if we get this particular color change. What does the L stand for in this acronym? Enzyme linked. What is that enzyme linked to? That enzyme is going to be linked to an antibody. So that's where the antibody comes in. So let's take a look and kind of run through the sequence of events that is involved in doing a ELISA test. Up here, this shows you a microtiter plate in which several different serological tests are being run simultaneously. Probably much like the viral hemagglutination test we showed you earlier on. This could represent several different patient samples, for example, and then probably several dilutions of that particular patient's serum. Okay. Yellow would indicate a positive result. Clear would indicate a negative result. So what's going on here? Well, these particular microtiter plates can be purchased to do particular ELISA assays. And in the direct testing method, what we're looking for is we're screening a fluid, patient serum, let's say, for the presence of particular antibody. So what are we going to be doing when we do that test? Well, we're going to purchase an ELISA plate that has antigen that is specific for the antibody we're looking for, physically attached to the inside of the wells of those plates. So if this is an HIV test, for example, you would buy a plate that had HIV antigens secured to the inside of the well. Okay, makes sense. So what do? We then pipette serum samples or dilutions into those particular microtiter wells. And if there is antibody present in that serum sample, it will attach to the antigen that is fixed to the well. And if there's no antibody present, it won't. Okay? After we do that particular step, we then wash out the wells to remove any unbound antibody. So if there were antibodies there, like this purple guy right here, that weren't specific for the HIV antigen, they wouldn't bind. And therefore they would be washed out of the well during the washing step of the ELISA reaction. But if there were HIV antibodies, they would fix to that particular antigen that's coded onto the well, and they wouldn't be washed out. They would be bound. They would be immunoabsorbed to the sides of that well, and they would be fixed. Okay? So the next step now is how do we know? We can't really see whether the antibodies have been fixed or whether there are no antibodies there. We need an indicator. So what do we add? We add that phosphatase enzyme that has been conjugated or chemically bonded to an enzyme. To an antibody, sorry, not an enzyme. And those antibodies are anti-antibody molecules. They will bind to the FC region of any antibody that has been absorbed to that well. And then we wash again. So any conjugated enzyme that did not have an antibody to attach onto would be washed away. In this particular case, because antibodies were absorbed to the wells, then the enzyme attaches to those bound antibodies, and it's fixed, even after we wash it. Okay. Last step is what? Add the substrate, add the NPP, and give the enzyme a chance to catalyze the reaction, which would result in a color change in that particular well. In this particular negative test over here, the enzyme would have been washed away. Therefore, when you add substrate, there's nothing for the substrate to interact with, and therefore no color change. So a positive test would indicate the presence of antibody in that particular situation. How many antibodies have to bind to this particular well? As few as one. Because if that enzyme is captured there, and you provided substrate, that one enzyme could just keep recatalyzing that reaction. And it might take a while to get a color change, but as little as one antibody molecule there's one trapped antibody molecule trapping one enzyme, could give you a visible reaction over time. So one of the things probably that's indicative of antibody amount that's been trapped is how fast that color change occurs. Okay, if it's really, really slow, probably very few antibody molecules are present. If a lot of antibody molecules were present and a lot got trapped onto that well, a lot of enzymes would have bound and the reaction would have produced a greater color change faster. Pretty inventive. In the indirect method, we are looking for antigen, so you buy a different kind of plate. You buy a kind of plate that has antibody affixed to it that is specific for that antigen. You add your test sample, and if the antigen matches the antibody, it'll attach, and then when you wash the plate, it won't be washed away. And then you add an enzyme-linked antibody that also attaches to the antigen. So the antigen kind of forms a sandwich between two antibodies. Wash again, add your substrate. If it turns yellow, then that would indicate that this particular antigen had been captured in the well, and we get a positive response. If not, we would get a negative response. So a very powerful method to very quickly, very accurately, and very sensitively detect the presence of antigens or antibodies in solutions that you are screening. Another method that's being used increasingly is serological tests are Western blots. Dr. Gordon McCombs does a lot of Western blotting in her biotech class and also in her research. The idea of blotting is to separate materials by electrophoresis on a gel, and then once that separation is taking place, to transfer the material that was in those bands on the gel to a blotting material, nitrocellulose. And then you can take that nitrocellulose paper and expose it to various screening reagents to see if the substance you're looking for is present or not present. So you can use ELISA, for example, in conjunction with Western blotting. Or you can use radioactively labeled antibodies. Or you can use chemoluminescent antibodies. The first blotting procedure that was developed was a blot called the Southern blot. And Southern blots were used to separate DNA molecules from one another on gels, like you did in cell biology. And then you could transfer that DNA from the gel into a blot and do some kind of detection method. Later, the technique was modified to separate RNA molecules and detect RNA molecules. Why was this called a Southern blot? This was called a Southern blot after the person who developed that particular technique. Why do they call this a Northern blot? Well, this was getting a little kind of cutesy now. They said, well, if we have a Southern blot, then RNA should be a Northern blot. They've also developed later this technique for separation and detection of proteins. And they continued their cuteness and said, well, if we've got Southern and Northern, we now have Western. Okay? So these particular blotting techniques are used very routinely now to separate molecules, and then other methods can be used like ELISA to identify the nature of those molecules. A lot of times blotting is used to verify positive results from other tests. So if you get an initial positive reaction from an ELISA screening for HIV antibody, that's a pretty serious diagnosis. You're going to want to confirm that. And confirming it by other methods like Western blotting, for example, and looking for specific HIV proteins in that sample is good practice. Okay, the last topic we want to talk about a little bit is monoclonal antibodies. One of the things that I said was important in serological testing is that there be a high degree of specificity and sensitivity. And you might be thinking to yourself, well, just kind of inherent in any serological test is a high degree of specificity because particular antibodies only react with particular antigens. That's where the specificity comes from. But we'll see in a second, it isn't quite that straightforward, particularly when you start to dig into a little bit, where do we get these antibodies we use in serological testing? Where do we get those antibodies we link to the enzyme in ELISA, for example? Where do they come from? How do we produce them? Okay, so what are monoclonal antibodies? So a little, definitions and things up here. Monoclonal antibodies are monospecific antibodies. They only react with a particular antigen. And the reason that all monoclonal antibodies are specific for the same antigen is that they're produced by the exact same type of immune cell. They come from a single clone of immune cells. That's what we call them, monoclonal antibodies. But remember, immune responses are notorious for giving you not a monoclonal response, not only a single lymphocyte is activated. It's not uncommon at all in immunology to get a polyclonal response. Several different antibodies are elicited by one molecule. Why is that? Why does one molecule potentially produce different types of antibodies against that molecule? Answer that question in probably 2 or 3 words. Because molecules have multiple epitopes or antigenic determinants, remember? Remember when the lymphocyte is responding to a complex protein, it's not responding to the whole protein molecule, but just a little piece of it. So let's say that you wanted to develop a test for the substance HCG. You know what HCG is? Physiology? Human chorionic gonadotropin. What's a common name for HCG? It's a hormone.
It's released when you're pregnant.
It's a pregnancy hormone, right? And so an antibody that could detect the pregnancy hormone would be a great basis for a pregnancy test, right? And that is indeed the basis of over-the-counter pregnancy tests are antibodies that detect the presence of HCG. But HCG is a protein. It's a complex molecule that has multiple epitopes or antigenic determinants. So if I took a mouse and I injected HCG into it, would it mount an immune response against HCG? Yeah, that's foreign to it. Okay, that's human chorionic gonadotropin. But what kind of a response would it give you? It would give you a polyclonal response because one epitope would stimulate one type of lymphocyte, and it would produce a particular antibody. But a different epitope on HCG would stimulate a different lymphocyte and give you a different antibody. So we would tend to get a polyclonal response. And that interferes a little bit potentially with specificity. It gives you some additional possibility for false positives. Because remember, sometimes antibody molecules can cross-react. So 2 different proteins and different organisms can both interact with the same antibody. That group A strep antigen and also human heart valve antigens, those heterophile antigens. So it really would be nice if we could have just a single antibody instead of a mixture of antibodies as the basis for our test, because then you'd be much more confident in your results. How do we get a single type? How do we get just one of these clones instead of dealing with all of these clones and all of the antibodies that they could produce? Well, there are techniques available for developing and producing monoclonal antibodies. And basically, you can produce monoclonal antibodies that will specifically bind to pretty much any substance that exists. And this has really become a very powerful tool in biochemistry, in molecular biology, and in medicine particularly, to both detect and treat diseases. And that's really particularly important. When we talk about monoclonal antibodies, we will give you some information about how they can use for disease detection, but also disease treatment. Now, I put the word disease in quotation marks because pregnancy is not a disease, although sometimes, after you watch your kids grow up, you might wonder. So, but this idea of detection and treatment is a very powerful set of outcomes that you can use in monoclonal antibodies. When you start looking at drugs, you can tell that a drug is a monoclonal antibody-derived drug because its name ends in MAB. And we'll show you some different monoclonal antibodies in just a minute. So, there's a variety of academic and medical and commercial uses for monoclonal antibodies. You can detect small amounts of drugs. You can use them in drug testing. You can look for toxins. You can test for hormones like the pregnancy hormones. You can use it to treat viral diseases that previously had been considered untreatable. You can identify and trace specific molecules in an organism. Monoclonal antibodies have been very powerful tools in further understanding of developmental biology. They've been really great in helping to alleviate the problem of organ rejection. In the past, before monoclonal antibodies were widely used, the method we typically used to try to suppress organ rejection was steroids. And what's the problem with using steroids? It's a real sledgehammer, because steroids basically depress your entire immune system. And if you've just had transplanted tissue or on steroids, you're trying to give your body time to accept the transplanted tissue, then you have to be put in isolation so you don't get exposed to things your immune system can't fight off. But what should we really be trying to knock out? Do we want to knock out the humoral immune response? Is that part of tissue rejection? No, it's a T-cell response. It's a cell-mediated immune response. So if we could develop drugs that would specifically suppress temporarily C cytotoxic cells and not harm T helper cells, that would be great. You'd have far fewer side effects from that particular treatment. And they have developed particular drugs that target specifically C cytotoxic cells and use those in place of steroids for treating people who have had transplantations. You can also use it in immunodetection and immunotherapy of a variety of cancers. And that's also been a very powerful tool. So, here's how you produce monoclonal antibodies kind of overall, and then we'll get into some little more details about this. So, the first thing you have to do is you have to take a test animal like a mouse and challenge its immune system with some antigen. Okay, in this particular case over here, the HCG molecule. So we want it to start forming an immune response against that. And it's going to do that, but it's going to give you a polyclonal response. It's going to produce many different clones producing many different antibodies. We want a single clone producing a single antibody. So what can you do? Well, you can harvest the spleen of that mouse after a while, because that's where a lot of the immune cells are going to be. Are there techniques where you can actually take a mixture of cells and sort them from one another based upon subtle cellular differences? Actually, there are. There are cell-sorting techniques. So could you go in and say, I'm only going to take out clone A, and I'm not going to take B or C or D or whatever. I just want to take clone A. Yeah, theoretically you could do that. But then we would want clone A to divide and grow to form more cells of that type to produce that antibody. Unfortunately, lymphocytes don't grow well on culture media. Lymphocytes, like a lot of other cells you grow in culture media, are mortal. What that means is that they only divide a few times before they naturally stop dividing. Almost programmed into those cells is signals that say you're only going to divide 20 times and you're done. That doesn't do us much good. What kind of cells tend to be immortal in terms of the number of times they can divide? And they keep growing and growing and growing. Cancer cells. Yep. So actually, in the development of monoclonal antibodies, in addition to lymphocytes, we use some cancer cells because of their immortality. And the type of cancer cells we typically use are myeloma cells. They're basically cancerous lymphocytes. So there are special strains of mice that have been maintained that have multiple myeloma. And we can harvest the lymphocytes from those and grow them in culture, and they thrive. They grow just fine. Unfortunately, those lymphocytes that have become cancerous don't produce functional antibody. So they're immortal, but they don't really produce the antibody they normally would. So myeloma, no functional antibody. So man, we've got kind of two situations here. We've got a lymphocyte that produces functional antibody, which is mortal, and then we've got a myeloma cell which is immortal but doesn't produce functional antibody. What we would like is a cell that is immortal and produces functional antibody, right? So what do? We take those two cells and we fuse them together. And we form a hybridoma, a hybrid cell. And that hybridoma secretes functional antibody, but is immortal. So when we do this fusion, remember we had several different lymphocytes that were produced. We had a polyclonal response. Each one of those theoretically could fuse with a myeloma cell. So we can produce several different hybridomas. We only want one. So what can we do? We can actually sort those out from one another and put them on culture medium. But now, because they're hybridomas, they're going to grow. And then it's a matter of screening each of those hybridomas for the best antibody for the particular purpose you want to use. The one that gives you the most sensitive test, for example, for human pregnancy. And we take that particular hybridoma, and then we just maintain it, and it keeps secreting that one monoclonal antibody. Now, are all of these fusions successful? No. There will be many situations where a lymphocyte won't fuse with a myeloma. So initially here we will get several hybridomas, but we're going to have unfused lymphocytes, we're going to have unfused myeloma cells, is there a way to kind of screen for successful hybridoma formation? And the answer is yes, there is. Okay, so let's take a look at this. So, take a normal mouse and we inoculate it with antigen like HCG. We're going to get a polyclonal response. We isolate those particular lymphocytes from the spleen, but they're mortal. We then take a special strain of mouse that has the cancer and we isolate the myeloma cells from one another. So then we fuse those two together to form functional hybridomas. But remember, many of these won't fuse. So we're going to have a mixture of many hybridomas, many lymphocytes, and many myeloma cells. There's a way to select for hybridomas only. And that involves a special enzyme called HAT media. And I don't care that you know that HAT stands for hypoxanthine aminoterine thymine. But basically, it's a selective media for hybridomas only. Lymphocytes won't survive, and myeloma cells won't survive. Only hybridomas will survive. It has to do with the presence of this particular enzyme, HGPRT, hypoxanthine guanine phosphoribosyl transferase. You don't have to know that name either. But basically, we've got this media, this HAT media we can use to select for only successful hybridoma formation. So we've eliminated the confusion of the unfused cells complicating our search for hybridomas. So here's our culture of surviving hybridomas. Each one of those is going to produce a slightly different antibody because they were responding, the lymphocyte was responding to a different epitope on that HCG. We can separate out these hybridomas from one another and separate them into individual wells. And then we can do screening for the particular antibody that each of these hybridomas produce and find the best, most sensitive antibody for that particular test that we've envisioned. And then we just pull that particular hybridoma off the plate. And we can then maintain it in culture because remember it's immortal. But as a backup, we typically inject that hybridoma into a mouse and let the mouse maintain it in vivo. And then we let that hybridoma produce the particular monoclonal antibody that that particular cell produces. So now we only have a single antibody molecule being produced, and then we can use that in highly sensitive and specific tests. It's a little video if you're interested to embed it in the PowerPoint that shows you how to make monoclonal antibodies. So here's a good practical example of monoclonal antibody testing. This is a rapid strep test. So when you go to the doctor now, instead of having to take a culture, put on blood agar, wait a couple of days to get growth, see if it's indicative of strep, if it has the right hemolysis before you can be treated, you can do this rapid strep test, and we're looking for the presence of particular antigens or antibodies, depending upon the nature of the test, that are associated with strep or strep infection. And typically there is a little colored reaction that takes place, in this case a little red stripe, that indicates a positive test for that particular organism. In this particular test right here, we're using monoclonal antibodies against a particular strep antigen associated with that pathogen. So this one has the antigen, this one doesn't have the antigen. Monoclonal antibody therapies. Monoclonal antibodies are kind of the biological smart missiles that can deliver very precise strikes to cancer cells or virus-infected cells and leave normal cells alone. How do they provide those precise strikes? Because if you can engineer monoclonal antibodies against particular cancer antigens, then you can conjugate to that monoclonal antibody a cytotoxic drug, if the monoclonal antibody only binds to the cancer cells, the cytotoxic drug will only be delivered to those cells, and it's going to leave normal cells unharmed. That's a lot better strategy than radiation, because radiation is very indiscriminate. The idea behind radiation is that radiation tends to affect rapidly dividing cells most, and cancer cells are pretty rapidly dividing. But there are other cells in your body, like epithelial cells, which are pretty rapidly dividing as well. And that's why you get all of the nausea and vomiting things, is that you're destroying epithelial cells with your radiation treatment. Or if you just bathe the body with a chemotherapy drug, it can affect non-cancerous cells as well as cancerous cells. But man, here, if you can have the smart missile with a monoclonal antibody that probably has been formed by extracting your tumor cells and using those as the antigen to make the monoclonal antibody, then you've got a really precise guided missile that can deliver the drug to your tumor cells. That's really very powerful. There are some problems with monoclonal antibodies. The first monoclonal antibodies that were produced in mice. Those are called murine monoclonal antibodies. Problem is if you start injecting mouse monoclonal antibodies into humans, you can get an immune response against the monoclonal antibody. Later on, they developed technology to make what they called chimeric monoclonal antibodies. They had some elements of the mouse antibody structure, but some parts of it were human. And now they've developed what they call fully humanized monoclonal antibodies, which are entirely human in nature and should not give you that kind of immune response against that introduced material. Even been some work to produce monoclonal antibodies in plants. The idea was that you could provide a monoclonal antibody therapy by introducing it into a banana, and you could actually take your monoclonal antibody by eating a banana. So there's really been a lot of advancements made in this particular area of kind of rejection of the monoclonal antibody. A lot of monoclonal antibodies have been developed to try to treat various types of cancer. And there's all kinds of these that are in the process of various stages of development and FDA testing. Here are some kind of strategies that monoclonal antibodies are being used for in terms of trying to block cancer. One way is to kind of target cancer cells so they're more visible to your immune system. So like the kick me sign, basically. Almost enhance opsonance. Another strategy that's had a lot of success is monoclonal antibodies that will block growth signals. Because cancer cells typically have to receive signals to allow them to continue to grow immortally. Oftentimes that's response to growth factors. So if you can block those signals, you can arrest cancer growth. Remember from cell biology, the cancer warrior video? Saw it right before spring break, probably. Angiogenesis, blocking blood vessel growth to stop tumors. And originally, the chemicals that were used that were anti-angiogenesis kind of affected the whole body, but now if you could have monoclonal antibodies deliver those drugs, they're much more effective. Delivering radiation to particular cancer cells. So instead of affixing cytotoxic drugs, you could affix a little radioactive molecule that would be delivered directly to the cancer cells. And then I also talked about delivering chemotherapy to cancer cells. So a lot of promise here. This is really a very active area of research in cancer treatment. I just put this in here because these are some anti-cancer and anti-inflammatory, you'll see later, monoclonal antibodies. And obviously you don't have to be responsible for any of this particular information. But you'll notice that the chemical name of these all ends in MAB, indicating these are monoclonal antibody drugs. This is one that's being used a lot. This is particularly blocking a receptor for a growth factor in breast cancer cells. This particular drug goes under the trade name Herceptin, and Herceptin is being widely used in certain types of breast cancer, those that have the HER mutation particularly. But there are also other growth factor inhibitors for certain monoclonal antibodies. Some of these monoclonal antibodies work to kind of stimulate lymphocytes. Various cytokines can be targeted by monoclonal antibodies. These happen to be some anti-inflammatory that are used to treat diseases like Crohn's disease, rheumatoid arthritis. Notice here there's some anti-rejection drugs here that block interleukin receptors on particular T cells, like T cytotoxic cells. Here's some just kind of other drugs that deal with viral infections. In this case, RSV is a virus that's pretty nasty for children. This is an anticoagulation drug right here. So again, this just kind of summarizes what some of the commonly used monoclonal antibody-based drugs are right now. And this is just a fraction of the list that's being used right now. Look at breast cancer treatment, colorectal cancer, leukemias, asthma, organ rejection, Crohn's disease, again, a viral infection, RSV. So there's really a tremendous amount of upside potential to the use of these particular monoclonal antibodies. Kenzie and Annie have been involved working over at B2S Lab with some monoclonal antibody research, and they're using that for some proprietary tests to look at different types of methods to detect, what we got, insulin? Yeah, so they're doing some diabetes research over there, and monoclonal antibodies are a big part of what they do. So for your last exam, it will be like the other exams with probably one slight difference. Given the short turnaround time that I have for getting grades in, I think probably what I'm going to do is only have multiple choice and some essay, and I probably will not have agree and disagree this particular time. So I'll probably stick with the 3 essay questions and the 8 to 10 point range, and then the remainder will be the multiple choice questions. PowerPoints are all posted for those. Really, it's the third line of defense, and then it's the serology up through the information that we've had today. So basically 2 study guides, 2 sets of lecture notes, and 2 PowerPoints. So Friday, when you come in, I will have at your tables a checkout list. And the checkout list basically is to get you to make sure that everything, excuse me, that's supposed to be in your drawers are. And then I will also have you clean your microscope and kind of wipe off the lenses and everything. And then remember, your known pictures are due end of day Friday, 4 o'clock, along with your unknown. And then the last thing would be is that any optional points would be due by end of day on Tuesday. And so that could be any ID factors. Anybody doing the Unseen Life on Earth? I'm just curious, the videos, some of you are. Okay, so those would be due. I think that's all really the kind of optional points that are out there. I sent you back your Making Medicines results and the optional points and score you got on that. So I'll work on the, when you turn those in, the urine samples and your unknowns on the slides. See how much of that I can get done. Okay? We'll see you tomorrow for wrap up.
Thinking about it for a second. Some people,